ton. Today we have Dr. Steph from Fatty 15 on the podcast, and we did talk with her last year. It was a huge success. People loved knowing more about this incredible nutrient called C15. And she's got a lot more updates, a whole new book out called The Longevity Nutrient. Hi, Dr. Steph. Great to see you.
Hi, Dr. Sanji. Wonderful to see you again.
Yeah.
Just the research around C15, over 100 peer-reviewed studies, with the longevity nutrient, the book that came out earlier.
Yeah.
So the book covers, this amazing, surprising and accidental discovery of C15, which is also called pentadecanoic acid, an odd chain saturated fat, right? And so the discovery that we made accidentally in dolphins, right, while taking care of older dolphins, we go do a deep dive, forgive the pun, into that story of the relevance of this nutrient, right, emerging as the first essential fatty acid to be found in over 90 years since omega-3, and really covering the science.
Yes, but maybe your background and how you were involved in dolphin research at the very beginning, and how you got acquainted with C15, because you're right, before you, I had never heard even of this fatty acid.
Absolutely. And that was the whole world. I mean, really kind of the same with even experts in fatty acids, the same space. So, so hi, everyone. I'm a veterinary epidemiologist and was recruited by the Navy 24 years ago to help continually improve the health and welfare of aging Navy dolphins, which was wonderful. I thought I would do that for a couple of years, and it ended up being a couple dozen years. And it's really an incredible population. The Navy has cared for this sustained population of about 100 bottlenose dolphins for over 60 years. And because of their great care, Navy dolphins live a lot longer than dolphins in the wild. So dolphins live to about 20. At the Navy, they're living into their 40s, 50s. And I went to go visit the crew just last week, and I learned that 2 dolphins this year are going to turn 60 years
old.
God.
So it's like that in human years would be like a 150-year-old dolphin.
So amazing. And so
what's the limit? What do we think?
They're pushing it. We're gonna find out. So
do these 60-year-old dolphins are metabolically very old?
So it depends.
They're looking really young here.
Yeah, well, and that was the whole interesting finding. So I was brought in to understand aging dolphins, because there's no knowledge of it, because they don't typically live that long in the wild. So what we found over a 10-year period, over a decade of studies, is that about 1 in 3 older dolphins develop insulin resistance, high cholesterol, chronic inflammation, fatty liver disease, right? This is all sounding very familiar territory. And as we just published a couple weeks ago, even the full suite of changes consistent with Alzheimer's disease. So we were able to leverage this advanced technology called metabolomics to look at thousands of small molecules present in their archived serum, as well as their all fish diets, to see which small molecules predicted the healthiest aging dolphins. And that's where we thought it would be omega-3s, because all they eat are fish. But instead, it was C15, this previously unassuming 15 carbon saturated fatty acid that was the top predictor of healthy aging dolphin.
How much of that was attributable to C15?
Well, absolutely. So we found, right, so these metabolomics, what it does is it ranks molecules. So it says, okay, we're going to take your top, first of all, is there a difference, right, between these two populations, healthy versus faster aging dolphins, right? And then what they showed was yes, there is a difference. And again, it had, this particular panel was unbiased. So it included lipids, carbohydrates, proteins, amino acids. It wasn't just fatty acids. And so it was this untargeted metabolomics, which is really the gift of this technology. And it showed the top 30 predictors of healthier aging dolphins. And at the top of that, so there were other molecules, to your point, it wasn't just it was only C15, but C15 was the highest ranking that was a nutrient that comes from food. So we clinged onto that because it's this is something we can actually have an intervention with. And so we then did we did a feeding study where we gave dolphins fish with higher C15. So we learned that some fish had no C15, while other fish that they were eating had higher amounts of C15. So we were able to, they're given 5 different types of fish that they can eat. Some had C15, some didn't. So we had done this experiment over, a 15, 20-year period where some dolphins were getting a low C15 diet, others were getting high. So we put a group of dolphins on a high C15 via high C15 fish diet. And that's where we saw complete resolution of things like anemia and improvement and normalization of triglycerides, cholesterol, markers of iron overload. And then and C15 was the top independent predictor of specifically of resolved anemia. So once we had that, we then moved pure C15 into the lab to then say, okay, can C15 stand on its own and not just be association or, again, like you're mentioning, being mixed with other potential confounding factors.
Mm-hmm. Okay, a couple of questions. How did you even come across the idea that it could be C15?
It was luck. I mean, so we started with a fatty acid panel because we went with a biased hypothesis, which you're not supposed to do, which is it was going to be omega-3s. And so, but we said, listen, let's just do a whole fatty acid panel and not just the different types of omega-3, omega-6s. We'll do a whole fatty acid panel. The panel that we used, if we had done the study 3 years earlier, Dr. Sanjeev, C15 wouldn't have been on the panel.
Wouldn't even on the
and
panel.
so if we had done the study 3 years earlier, this discovery would not have been made. So we're lucky that, and it's also a big reason of why C15 is like, why wasn't this discovery made before? That for a long time, C15 has been used as the reference fatty acid. So almost like the dime that you put next to other things to understand the size of everything else. It was the reference fatty acid to measure all the other fatty acids. So by default, it was excluded as being evaluated as, does it itself have benefits? So once fatty acid panels started including C15, which was again, 3 years, before we started the study, that's when it started showing up in human studies too. So there are now multiple meta-analyses. So studies of large-scale studies, prospective cohort studies, following tens of thousands of people over a decade or more, showing just like we found in the dolphins, people with higher C15 have a lower risk of developing type 2 diabetes, coronary heart disease, heart failure, and specific types of cancer. So once we saw those parallels, then we said, gosh, let's go and move this molecule into the lab and see what it can do.
Oh, so these human studies, observational studies have existed for some time is what you're saying. People are already been looking at this. Is that what you're saying?
Exactly. So it was in the literature and it was emerging at the same time that our studies were coming out. Again, because now fatty acid panels were just starting to include C15. So, but interestingly, those studies, they focused on the fact that even chain saturated fats like C16 and C18, so have an even number of carbons, these same studies showed that higher levels of C16 or associated with a higher risk of type 2 diabetes, fatty liver disease, heart disease, which fit our
thinking
mantra, right? Yeah. So it's like, so even though, and then what they have is at the bottom, they said, well, so exactly what we thought, higher saturated fats equals bad health. That's and right. Then it was almost like a postscript. Oh, and by the way, we found that these trace odd chain fatty acids, that they were higher, interestingly associated with the opposite, better, but really let's just focus on the fact that we proved our hypothesis.
Okay. Interesting. What type of range is there for C15?
In general, the global population is sitting around an average of about 0.2% of our fatty acids are C15.
So very relatively low. If you look at omega-3, that's like 5% or something. Is that correct?
Exactly. People, in the fatty acid world, like, well, it can't be important. It's present at such low levels. And, what we've now understood is that we just need enough, especially of C15 to activate certain receptors, but also to stabilize our cell membrane. C15 is a stable fatty acid that its job is to help stabilize our cell membranes against lipid peroxidation. We don't need a lot, but if we don't have that minimal amount, then the cells become fragile, more susceptible to lipid peroxidation. And in fact, what we've been able to show is that if cell membranes don't have enough C15, it causes ferroptosis, which is an entirely new form of cell death that was discovered by researchers at the Columbia University back in 2012. the same time that we reported this phenomenon in dolphins, we just didn't know that it had a name. So a nutritional deficiency.
You mean death by iron? What does that mean?
Yeah, that's exactly it. So what they found, it is a very distinct picture. And this is a big deal, right? Because, we go to medical school or you go to cell biology class and there are 3 ways that our cells die, right? It's apoptosis, necrosis, and autophagy. Mm-hmm. And this crew at Columbia University found a 4th way, an entirely new way our cells were dying. And when they looked under the microscope, they said the cell membranes were thickened, the cell was intact, and the mitochondria were all dead, and there was iron inside. So it was a very distinct picture.
Right.
And then the way they described it was literally the fatty acids in the cell membrane were fragile. It led to lipid peroxidation. That lipid peroxidation then combined with this free iron that was sitting in the cell results in massive reactive oxygen species, took out the mitochondria, kills the cell. So since that paper, there have been over 10,000 papers on ferroptosis, which I had never even heard of. And we now know that ferroptosis accelerates aging. It accelerates the onset and progression of type 2 diabetes, heart disease, fatty liver disease. What nobody could answer was, where did this come where did this come from? And that's where the dolphins gave us, this intel, which it popped up in dolphins because of C15 deficiencies. And we were able to fix it by putting C15 back into their diets.
So do we think that iron itself, like these people are susceptible with low C15s, giving iron supplementation may not be helpful? Is that?
Yeah, it's this huge question right now, right? So because, and you're spot on, because what happens is, we've now been able to describe the whole pathophysiology of this, what we're calling cellular fragility syndrome. We published in Metabolites last year. And what we now know is that our cells become more fragile. This includes our red blood cells. So as our red blood cells become more fragile, they get engulfed by cells within our liver called Kupffer cells. So they're these macrophages that then engulf weakened red blood cells. The problem is we have lots of weak red blood cells, right? That results in basically the corpses of these red blood cells being iron. And then you get iron overload in the liver. This is called dysmetabolic iron overload syndrome in people. And then that then feeds the beginning of ferroptosis. That then spills over systemically and this takes time, seeds the heart, seeds the brain, seeds the pancreas. And that then leads to, if you look up iron overload and these conditions, last year, 2024, there were 1,024 papers written on iron overload. This is just starting to hit the medical and science community, and understanding the way that you're seeing it in patients would be hyperferritinemia.
Yeah, that's what I was going to just ask about. Ferritin might be a very good marker. So potentially sometimes we see people just high ferritins, but we don't really know what to really make of that. But you're saying potentially C15 should lower that ferritin then.
That's right. And that's what we saw in the dolphins. We saw basic increases in liver enzymes, and we saw hyperferritinemia. And so when we then went back and looked at dolphin liver tissues, that's when we saw, and that's what we published in 2012, iron deposition in the liver, specifically in the Kupffer cells. So it was like we knew exactly what was happening. So what C15 is able to fix is stabilizing those red blood cells, right? Stabilizing all of our cells, but especially our red blood cells. That stops the whole pathophysiology of the disease by stabilizing the cells. Then you don't get iron overload. We're able to move this into a model, a controlled animal model that mimics this whole pathophysiology, and showed that by giving it C15, supplementing them, that it was able to completely stabilize the red blood cells, stop the iron deposition in the liver and the downstream complications. So, but once the iron is in the liver, though, C15 can't pull it out. So it will stop the progress. What we did with the dolphins is we used phlebotomy, right? A classic old method for getting iron out of the tissues. You draw blood and the iron comes out of the tissues. But then once they had the proper C15, then, the problem was resolved.